Hello everyone, this is Tejasvi. Today I'm gonna explain you about gene cloning. In this session, I will let you know what is gene cloning, steps involved in gene cloning, and types of restriction and modification systems, and finally the naming of restriction endonucleases. First, let's talk about the gene cloning. Gene cloning is the process in which a gene of interest is located and copied out all the DNA extracted from an organism. First, a fragment of DNA contains a gene to be cloned and is inserted into a circular DNA molecule called vector to produce a recombinant DNA molecule. Basically, there are 5 steps involved in gene cloning. First, construction of recombinant DNA molecule. This is the vector and this is the gene of interest. When we combine vector and gene of interest, it forms the recombinant DNA molecule. Let's come to the second step. The second step involves the transport of recombinant DNA into a bacterium. The vector transports the gene into the host, which is usually a bacterium, although other types of living cells can be used. And the third step involves multiplication of recombinant DNA molecule. Within the host cell, the vector multiplies, producing numerous identical copies, not only itself but also the gene that is carried. And the fourth step involves the division of the host cell. When the host cell divides, copies of the recombinant molecules are passed to the progeny, and further vector replication takes place. And the fifth step involves the screening of recombinant DNA. After a large number of cell divisions, a colony or clone of identical host cells is produced. Each cell in the clone contains one or more copies of recombinant DNA molecule. The gene carried by the recombinant molecule is now said to be cloned. Now let's talk about the types of RM systems. RM means restriction and modification systems. There are 4 types of RM systems: type 1, type 2, type 3 and type 2S, which differs primarily in structure, cleavage site, specificity and cofactors. 4 different types of restriction and modification systems have been recognized, but only one is widely used in gene manipulation. Type 1 restriction and modification system have active enzymes consists of subunits. The subunits are recognition, cleavage, and methylation. Recognition and methylation methylates a single sequence but cleaves DNA up to 1000 base pairs away. The typical example of type 1 is E. coli. 2 restriction subunits, 2 modification, and 1 recognition subunit. These subunits are the products of HSDR, HSDM, and HSDS. The methylation and cutting reactions both requires ATP and adenosyl methionine as a cofactor. These features mean that type 1 systems are of little value. Now let's talk about the type 2 system. The type 2 system have 2 different enzymes which recognizes the same target. Most of the useful RM systems are of type 2. They have a number of advantages over type 1 and type 3. Restriction and modification are mediated by separate enzymes, so it is possible to cleave DNA in absence of modification. Restriction activity do not require any cofactors such as ATP and S-adenosyl methionine, making them easier to use. Now let's talk about the type 3 restriction and modification systems. One enzyme with 2 different subunits cleaves 24 to 26 base pairs away but recognizes and methylates the same sequence. Enzymes have symmetric recognition sequence, otherwise resembles the type 1 system. Now finally talk about the type 2S restriction and modification system. Although the type 2S system have similar cofactors and macromolecule structures, the fact that restriction occurs at a distance from the recognition site limits their usefulness. Today's final topic is the naming of restriction endonucleases. The naming of restriction endonucleases provides the information about their source. A suitable system of nomenclature was proposed by Smith and Nathans. For example, Eco R1. In Eco R1, the first letter E represents the genus, and the next two letters CO and represents the species, and the final letter R represents the strain, and the one represents the order of discovery. The Eco R1 full form is Escherichia coli. R is from strain R by 13, and 1 is as it is the first enzyme to be isolated from the given strain.